I was introduced to the topic of colony collapse disorder during my undergraduate studies at UF. At that time, in 2006, beekeepers and scientists were still unaware of what was causing the disappearance of the honeybees. Honeybees are overwintered in Florida, then shipped across the country to pollinate crops, specifically like almond trees in California. When the pollination services were over, beekeepers reported over 50% in colony losses. There was no sign of the bees, they just simply vanished. Since then, many studies have been done detailing the causes of bee death, and more studies are currently underway on how to stop and reverse this disorder. One such avenue involves restoring the ecological connection between bees and mycelium. Let's start by spending a quick minute on honeybees and the details of colony collapse disorder, also known as CCD. Much of the information on this presentation pertaining to bees was taken from a class given by Dr. Jamie Ellis at the University of Florida. So we're specifically discussing Apis mellifera, also known as the honeybee. As you can see, there are specific symptoms that make up CCD, but you don't find evidence of additional pests or catastrophic diseases. One thing I'd like to mention is that beekeepers typically remove the honey surplus and then supplement the bees' food with a corn or sugary syrup, which I'll mention again later on. There are some factors scientists agree upon that have led to CCD. To make honeybees more robust against environmental conditions, disease, and to produce optimal honey products, bees have been bred into a sort of monoculture by injecting specific semen into queens that are then sold around the world. This has resulted in a severe lack of genetic diversity. Bees are also subjected to a loss of nutritional diversity as they are transported to specifically pollinate monoculture crops. This means they are lacking a well-rounded diet. Varroa mites are a bee parasite that sucks the bee blood, called hemolymph, and vectors viruses and other pathogens into the bees. They are a major contributing factor to bee decline and death. While there haven't been any major links to CCD and genetically modified crops themselves, the pesticides sprayed on these crops are no match for the delicate honeybee. Neonicotinoids are one such type of pesticide that further reduce colony genetic diversity. They decrease the bee's ability to fight out viruses and reduce the lifespan and sperm viability of the male bee. Finally are all the toxins bees come into contact with. They collect tree resin to create a substance called propolis, acting as a shield to seal their hives, and so will collect other similar sticky substances like paint and glue, which turns the propolis from protective to toxic. So here's where we bring in the mushrooms. We are currently experiencing what scientists have coined the sixth great extinction, or the Anthropocene extinction, where masses of floral and faunal species are going extinct because of the environmentally destructive activities of humans. Deforestation is the main cause of this extinction. Old-growth forests hold vast libraries of knowledge that are being clear-cut and burned, making way for monocultures, which drastically reduces fungal diversity. Now this is the cool part. Mycelium express extracellular droplets that contain unique blends of acids, enzymes, and messaging molecules specific to each species. They act as a natural internet, connecting plants and learning from their environment. Thousands of nuclei interact with their surroundings as they move through the forest. When they encounter something new to digest, a new toxin is expressed, and that knowledge is transferred genetically through the entire mycelium mat, enabling the fungus to learn as it grows. Bees self-inoculate themselves with these droplets. There are 5 types of mushrooms that I'll be discussing: the red-belted polypore, the amadou, the agarikon, the reishi, and the chaga. The red-belted polypore lives most commonly on dead coniferous trees like hemlock and spruce, mainly in old-growth forests. What's amazing about this mushroom is its ability to break apart those pesticides, as I mentioned earlier. Now, honeybees make a substance called bee bread, which is made from collected pollen and lactic acid regurgitated from the bees. Bee bread has beneficial fungi that are susceptible to fungicides in the environment. Bee bread also has a multitude of yeasts and bacteria that correlate to the genetic diversity of the bees themselves. So that means as we narrow the genetic diversity of bees through selective breeding, we're narrowing the microbes as well. That's not all. Mushrooms have something called polyphenol p-coumaric acid. This acid decomposes wood and controls the detox pathways that allow bees to remove toxins from their bodies. No decaying wood means no mushrooms, which causes the accumulation of toxins in the honeybee. The amadou mushroom was first described in 450 BCE for its anti-inflammatory properties. And then in 1960, it became the first antiviral substance known to medicine. It mainly grows on birch trees, but can be found on many others as well. Recall the varroa mite is one of the main pests of the honeybees. One of the main viruses vectored by the varroa mite is called deformed wing virus. This virus disables the bee's ability to fly and collect food for the colony. An elixir containing amadou mushroom extract has been shown to reduce this virus by more than 1,000 times compared to the control. The agarikon mushroom was first described in 65 AD as an elixir for long life. It's found exclusively in old-growth forests and now believed to be extinct in Europe and much of the world, where deforestation has taken down the older mother trees. Agarikon is the longest living mushroom in the world. Mushrooms are afflicted by many of the same issues that affect humans and bees. Therefore, we can utilize the same defense mechanisms against those same stressors or pathogens. As bees bring mycelia extracts into the hive, the bioactive substances are stored in the honey, bee bread, and propolis, allowing for social immunity and self-medication. Reishi mushrooms were first mentioned during the Qin dynasty. They were reserved for royalty and used to improve general health and extend life. 3 species have shown extensive antimicrobial activity. These graphs show the extracts of feeding reishi mushrooms to honeybees. Do you remember how beekeepers remove the honey and instead supplement the bees' food with a sugary syrup? Well, blue shows the control group fed this syrup. Red, green, and yellow show 0.1, 1, and 10% reishi solution respectively. The first shows lifespan. You can see how both the red and green lines are above the blue, showing consistent extended survival rates. Now the bar graph shows the same solution, but this time against viruses. The bees fed simple sugar have a significantly increased viral count compared to the ones fed the reishi mushroom extracts. Chaga is mainly found on birch and beech trees throughout the boreal forest. It grows on living or freshly cut trees, but as the tree dies, the active compounds from the mushroom decrease dramatically. This graph shows the viral burden reduction when captive honeybees were given a solution containing extracts from the chaga mushroom. Again, the blue bar references the control group of captive bees fed the sugary solution. Notice how the viral burden skyrockets compared to the chaga mushroom extract. The viral burden is drastically reduced. Agarikon, chaga, amadou, and reishi all show antiviral activity in Apis mellifera. So in conclusion, how do we restore the ecological connection between bees and fungi? And why is it so critical? Well, the answer is multifold. First, we need to stop artificially inseminating queens and allow them to choose their mate more naturally. Increasing genetic diversity in bees means increased microbial and fungal diversity within the colony and the ecosystem. Second, we need to allow bees to eat their own honey. Honey constituents not only upregulate detoxification and immunity genes in the honeybee, they also contain beneficial fungi and other microbes essential to honeybee health. And next, Humans need to stop applying so many pesticides, which includes neonicotinoids and other fungicides and miticides, etc. We also need to allow more mushrooms to grow that break down pesticides like the red-belted polypore. Number 4, and this is the big one. Mushrooms require many different trees as hosts, so by halting deforestation and working towards biologically diverse reforestation, we can maintain the genetic integrity of the fungal species within the greater ecosystem, allowing bees to self-medicate. As a recap, the reishi, amadou, and chaga mushrooms reduce viral loading in honeybees. The reishi and agarikon extend the bee's life, and the red-belted polypore assists with detoxification. We have barely scratched the surface of the mycelium host defense knowledge. I'd like to take a quick minute to thank both Dr. Jamie Ellis from the University of Florida and Paul Stamets from Fungi Perfecti for their ongoing research. Learning from them has been absolutely inspiring. This spring, Paul Stamets and his team at Fungi Perfecti will be releasing a combination of the mushroom extracts mentioned in this presentation to counteract many of the issues the honeybee is currently facing. I'll be working with the Mount Diablo beekeepers from around the San Francisco Bay Area conducting field tests. We'll compare the effect of the extracts to actual mycelium grown on stumps, kits, and in mulch piles. We may even be able to introduce mycelia plugs directly into the bee boxes. I'll be posting this presentation along with updates on how the bees react to the fungal reintroduction. I hope you'll join me.